Hello my dear students, I hope you all have been doing well. From today we are going to start our biochemistry course, and this is my lecture number 1. And first of all, we would be studying in this course about the protein, and in this week we would be studying about the protein. We would be studying about the function of protein, structure of protein, and how protein is made up of from monomeric unit that are amino acid. So first of all, my first lecture is based on the all the information about the amino acid. Actually, in this week we have to study about the protein, but before starting studying protein, we should have to first know about the amino acid, because protein amino acid are monomeric unit of protein, and protein are large macromolecules that are made up of amino acid. So to know about the structure and function of protein, we should must know about what are amino acid, and how amino acid means what is the structure of amino acid, and what are the chemical and physical properties of amino acid, and how protein how these amino acid assemble together and to form polypeptide chain and then the protein. So now let's start from here. So first of all, we have to know about what are amino acid. So actually amino acids are the organic molecules, and these are the monomer of protein, and then there are 20 amino acid or sometimes 21 that are present in the protein. So actually, what is the protein and what are the amino acid? Protein actually are the larger macromolecules, and these macromolecule are formed after the joining of the monomeric unit that we call as amino acid. And amino acids are, what are amino acid? Amino acid are organic molecules, and these are composed of amino group, carboxylic group, and one of the R chain, and one of the hydrogen atom that is present in all amino acids. So when we talk about the structure of amino acid, so the amino acid is having one amino group and one carboxylic group. All the amino acid would be having one amino group and one carboxylic group and one R chain and one hydrogen atom. So except the glycine is having two hydrogen atom. Glycine side chain is another hydrogen atom. So except the glycine, all the amino acid is having one amino group in its structure and one carboxylic group in its structure and one side chain that we can call as R group, and one of the hydrogen atom that is found to attach. All these groups found to attach with a central carbon atom that we say as alpha carbon atom. So one important thing that we should have to know about the all 20 amino acid. There are 20 amino acid or 21 that mostly 20 amino acid that exist in all type of protein, or they that occur in nature. These are the 20 standard amino acid. So all the 20 standard amino acid is having one amino group, one carboxylic group, one hydrogen atom, and one side chain that is the R chain or R group. So all the 20th amino acid is having different side chain. So due to the differences in the side chain of all the amino acid, so we are having different type of amino acid. So we will be seeing in the next slide what are the different type of amino acid, but up till now you have to just remember this thing, or keep this thing in your mind, that amino acid are the organic molecules, and these are composed of carbon, nitrogen, and hydrogen atoms, and these are having one central carbon atom, that is alpha carbon atom, and this alpha carbon atom found to attach with one amino group, with one carboxylic group, and one side chain, and one hydrogen atom, except the glycine, because in the glycine the R group is hydrogen atom. So what is the amino acid? Amino acid definition is, or amino acid is an organic molecule, or organic means you can say organic entity that is having that is made up of carbon, hydrogen, and nitrogen atom, and it is having one central alpha carbon atom that is found to attach with one amino group, one carboxylic group, one R chain, and one hydrogen atom. So this is the standard definition of amino acid, and as I have told you previously that all the amino acid we are having different amino acid, 20 different types of amino acid that are existing in nature, and the reason why we are having different amino acid, the reason is behind this, that all the amino acid is having different R group, that is the side chain. So we can see here we are having 20 different amino acid in the nature. And for example, if we talk about the glycine, that is one of the simplest amino acid. So we can see that this amino acid is having one carboxylic group, one amino group, and one side chain. And in the case of glycine, this side chain is hydrogen atom. So this is the alpha carbon atom, and this is attached with another hydrogen. So when we talk about the carbon, you know about this thing that carbon is having 4 valencies. So if we go back, so if we are having carbon in nature, so carbon is having 4 valencies. So what does mean by valencies? It means the carbon is having 4 free places that can be occupied with different atoms. So in case of amino acid, this carbon atom is having 4 valencies, so it is present in amino acid. So what happened in amino acid? This carbon atom occupied with one amino group, and other valency of this carbon atom occupied with carboxyl group, and another third valency of this carbon atom occupied with hydrogen, and the final valency, that is fourth valency of this carbon, is occupied with R group, that is side chain. So in 20 different type of amino acid, we are having different side chain. So one of the simplest example of amino acid is glycine, that is having another hydrogen atom as a side chain. And if we talk about the alanine, so we can see there we are having methyl group as our side chain, and in case of valine we are having another side chain. And all these type of amino acid, you can see that you are having same carboxylic group, same amino group, and same hydrogen atom that is found to attach with carbon atom, but all these amino acids are having different side chains. So all these are the 20 different type of amino acid, and that are existing in the protein. And all the proteins, there are different type of protein, and the proportion of these amino acid found to be different, and different type of amino acid. So now we talk about that how we classified the amino acid. Actually, amino acid classified on the basis of the differences in the side chain of amino acid, or especially the polarity of the side chain of these amino acid at the physiological pH. So we are having the amino acid on the basis of the polarity or on the basis of the properties of the side chain of the amino acid in the physiological pH, amino acid could be classified into 5 different types, and another classification of amino acid is based on its nutritional requirement. So first of all, we would be talk about how amino acid could be classified on the basis of their chemical properties of the side chain, or on the basis of the polarity of the side chain at physiological pH. So on the basis of the polarity of the side chain, so what is polarity? First of all, we have to understand it. So polarity is the ability of any functional group to make a hydrogen bond with water or with another atom, for example oxygen and nitrogen. So if a functional group or atom could be able to make hydrogen atom with another atom or with another functional group, so it means, or could be able to form the hydrogen bond with the water, so it means the molecule is soluble in water, or molecule is a polar. So here we are having different types of amino acid different classes of amino acid. So one is the non-polar amino acid, one other class is is having all the amino acid that are having polarity, means that are having side chain, and that side chain is able to make the hydrogen bond with water or other atom. And then one class of amino acid is the aromatic, and then other is positively charged and other is the negatively charged. So one by one we would be studying each class in coming slide, but you have to just keep in your mind here that amino acid could be classified either on the basis of the chemical properties of their side chain or the polarity of the side chain, and other classification is based on the nutritional requirement of amino acid. On this basis, the amino acid could be classified into essential and non-essential and semi-essential amino acid. So essential amino acid are those that our body cannot be able to synthesize, so we should have to take it from outside. So these are the essential amino acid. They can be isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan and valine. So the non-essential amino acid are those amino acid that our body is having ability to synthesize within itself. So our body could be efficiently synthesize the non-essential amino acid that we should not have to take from outside of our body or in our food. And another classification is the semi-essential amino acid. So these semi-essential amino acid are, we can say, conditional amino acid, means we just need to take it from outside of our body on the basis of requirement if we are deficit of these type of amino acids. So these are the two amino acids, that is arginine and histidine. So now we would be discussing extensively each class of amino acid. So first class is non-polar aliphatic amino acid. Or what are the non-polar or aliphatic amino acid? As I have already explained you in previous slides, that amino acid are classified on the basis of their characteristic of its side chain. So what is side chain? Side chain is R group. Side chain is R chain or R group. So we are seeing here one of the simplest amino acid, that is glycine. It is having one of the amino group, one of the carboxylic group, and one of the hydrogen atom, and one is the R group. And R group of glycine, that is one of the simplest amino acid, is another hydrogen atom. So glycine is one of the simplest and one of the smallest amino acid, because it is having smallest side chain, and that is hydrogen atom. So what is the aliphatic R group? Aliphatic means this class of amino acid. In this class, all those amino acids are listed that is having non-cyclic side chain except proline. So proline is having aliphatic side chain that is present in cyclic form, but this is aliphatic side chain. So and these class of amino acid is having all those amino acid that are non-polar. So what is non-polar? They are not having any functional group that can be able to make bond with the hydrogen bond with the hydrogen of water, or this class of amino acid is not having R side chain, R chain or R group or side chain that cannot be soluble in water. So here we can see that alanine we are having methyl group, and it is the also non-polar because it cannot be able to make the bonding with the water. So it is a non-polar. So we are here in this class we are having glycine, that is one of the simplest amino acid and smallest. Proline, that is having aliphatic side chain that is having that is present in cyclic form. So and this one of the important feature of proline is that it is having amino group rather than amino group. We can see here it is having H2 and NH2 group, not NH3 group. So this is the amino group and this is the amino group that all other amino acid is having. So from all the 20th amino acid, proline is only one exceptional amino acid that is having amino group rather than amino group. And from all the 20th amino acid, glycine is exceptional, that is having hydrogen atom as a side chain, and it is one of the simplest and smallest amino acid. So you have to keep in your mind uniqueness of these two amino acid. One is glycine and one is proline. And one more thing that I wish to explain you here, that these non-polar amino acid that are having aliphatic R group or side chain, so all these side chain are highlighted with the pink color. So all these non-polar amino acid, these are hydrophobic amino acid, means they are not able to have interaction with water. They are not hydrophilic, that are hydrophobic, means they repel water. Why they repel water? Because they are not having any side chain that could be able to make hydrogen bond with the water. So here we can see all these amino acid, leucine, isoleucine, methionine, valine, proline, alanine, all these are not having any charge, any polarity. So due to the unavailability of charge, they cannot be able to make the hydrogen bond with water. So these are the non-polar amino acid, and especially from these non-polar amino acid like alanine, valine, isoleucine and leucine, due to the inability to make the bonding with water, or due to their hydrophobic nature, these amino acid found to be buried inside the polypeptide chain or inside the protein. So what does it mean? It means that when these amino acid assemble together to form polypeptide chain, so you know the thousand amino acid assemble together. How they assemble we will discuss it later, but first of all you have to understand that when many amino acid join together, they form polypeptide chain. So many different polypeptide chain collect together or join together and form a protein. So in protein, for example, we are having a protein molecule. So in protein, if we are having alanine, proline, alanine, valine, isoleucine and leucine. So all these alanine, valine, leucine and isoleucine, if it is present in the protein, so they will be found to be buried inside the protein. Means these are hydrophobic amino acids, so they cannot be able to make a bond with water, They will be repelled by water, and they could be they will move towards inside of a polypeptide means protein. So what you understand what I am saying, that if you are having a protein and this protein is having a large number of these hydrophobic amino acid or non-polar amino acid like alanine, valine, leucine and isoleucine. So these amino acid, due to their hydrophobic nature, they would be found inside the protein, not from they will not found over the surface of protein. So that is what I wish I was willing to explain you, that you have to keep in your mind that all the non-polar amino acid that are hydrophobic in nature would be found to be buried inside the protein due to their hydrophobic nature. So this is this was the one important thing that I would wish to explain you. And one more thing, that one of the amino acid that is non-polar and having aliphatic R group, that is the methionine. So this amino acid is having thioether group, and it is one of the two sulfur containing amino acid. It is containing sulfur in its side chain, and other amino acid is existing in nature, that is cysteine. So cysteine is also containing sulfur in its side chain. So there are two amino acid that are existing in nature that are having sulfur group in that that is having sulfur atom in that side chain. So what are those amino acid? One is the methionine, that is having sulfur atom, and this sulfur atom is making thioether bond with other atom of the side chain. And one other amino acid that we will be studying in next slide, it is the cysteine, that is also having sulfur atom in its side chain. So this was all about the non-polar aliphatic R group, and now we will be studying about the the amino acid that are having aromatic R group or aromatic side chain. So what are these aromatic side chain? What is the aromaticity, or what is the aromatic? Aromatic means a molecule that is having aromatic ring. So what is this aromatic ring? Aromatic ring like a benzene ring. Aromatic ring is a ring or a benzene ring that is having conjugated double bond, and that is cyclic in nature. So here we can see that phenylalanine, tyrosine and tryptophan, these all are having side chain that are having a cyclic ring, that is a benzene ring that is having conjugated double bond. So on the basis of having this ring, we call these amino acid as the aromatic amino acid, or the amino acid that are having aromatic R groups. So one more thing that I would wish to explain you here, that the tyrosine is having hydroxyl group as a functional group, and the tryptophan is having indole ring in its structure. So this hydroxyl group and the hydroxyl group of tyrosine, due to having hydroxyl group, tyrosine take parts in various important biological functions in the cell, and the tryptophan is having indole ring, and this indole ring is also important for the biological activity of various protein and for the characterization of protein. So in our next slides we would be studying that what is the characterization of protein and why this tryptophan amino acid indole ring is important for the characterization of protein. But up till now you have to understand this: the aromatic amino acid are those amino acid that are having aromatic side chain. And what is this aromatic side chain or aromatic R group? The aromatic R group are those R group that are having the cyclic structure or cyclic benzene ring with conjugated double bonds. And these are three type of amino acid, that is phenylalanine, tyrosine and tryptophan. And the tyrosine is having hydroxyl group in the side chain that confers that confers it various important biological function, and the tryptophan indole ring in its side chain is also important for various biological activities of the protein. So another class of amino acid are those amino acids that are polar in nature, but they are uncharged. Uncharged means they are not having any charge. How these amino acid is, if they are not having any charge, could be act as polar, we would discuss here. So we are having what are the type of this polar uncharged amino acid. We are seeing here that we are having serine, threonine, cysteine, asparagine and glutamine. So all these are polar uncharged amino acid. So how these are the polar if they are not having the positive and negative charge, or they are not having any charge. So why they are polar? Because for example, in case of serine, we are seeing here the serine is having hydroxyl group. So this hydroxyl group is having capability to make hydrogen bond with the water. So water formula is H2O. So the oxygen of water will make hydrogen bond with this hydrogen of hydroxyl group. So by doing so, this serine would be act as polar, and it will be having some solubility in water. So in case of threonine, we are also seeing it is also having hydroxyl group that can make hydrogen bond with water. Due to this, it is polar. In case of cysteine, we are having sulfhydryl group here, SH, that would make bonding with water. So the cysteine is one of the second sulfur containing amino acid. So what was one? One was methionine. Here we see here in this slide that methionine is one of the sulfur containing amino acid, and second sulfur containing amino acid is cysteine. And one more important thing that I would wish to explain you here, that cysteine is one of that amino acid that can be oxidized, and when this cysteine oxidize, it form a dimer of amino acid, means it form dimeric amino acid within the protein. If we are having a protein and protein is having cysteine amino acid, so what would be happen then? The cysteine amino acid would get oxidized. This SH group of cysteine would oxidize, and this cysteine make disulfide bond with the other within the protein. So the cysteine amino acid after oxidation form a dimeric amino acid that we call as cysteine, and this cysteine is a dimeric amino acid that link two amino acid, cysteine amino acid, with a disulfide bond. So due to this property of cysteine, oxidative property of cysteine or oxidation property of cysteine, many polypeptide chain means linked together via disulfide bond. So we also would be studying it in our next slide, how cysteine get dimerized and make bond within polypeptide. But here you have to just keep in your mind that cysteine is one of the second amino acid that is sulfur containing. And when cysteine oxidized, cysteine has property of oxidize of the oxidation, and when it oxidizes, it form a dimeric amino acid that is cysteine. Now we will talk about the other amino acid that are polar uncharged. One of the amino acid is asparagine and one is glutamine. So these both amino acid, asparagine and glutamine, having amide group in its side chain, and these are the amide of two other amino acid that we would be studying in the coming slide, that are glutamate and aspartate. So here you have to keep in your mind that we are having asparagine and glutamine. These are polar amino acid, these are uncharged, and these are having amide group in their side chain. So now we would talk about the positively charged amino acid, or those amino acid that are having positively charged R group. So these are the lysine, arginine, histidine. So first of all, we would be studying, we would discuss about the arginine. Arginine is having guanidinum group. So this group is called as guanidinum group. And then lysine is an amino acid that is the positively charged amino acid, and this amino acid is having second primary amino group. So here we can see first amino group is here and second primary amino group is here at the epsilon position of this side chain, of its side chain. So how we wish to study here also, we can see here this amino acid is having charge positive, negative, and then positive. So why it is means not means neutral or not uncharged? Why it is charged amino acid? Why it is charged? Because one positive and one negative group cancel each other, and then we are having overall charge on the amino acid, one positive charge. So due to this positive charge, this amino acid is positive charge. And why we are having means although these amino acid, polar amino acid, having charge, positive charge on amino group and negative charge on this carboxylic group, except that why we are saying this uncharged, because this positive and negative charge will cancel each other. So overall we would be having no charge on the amino acid. So due to this we are saying that although the amino acid polar, because it is having the hydroxyl group that could be able to make the bonding with water, but this amino acid is uncharged, because although it is having the two charge, but this two charge would cancel each other. So here why we are saying that these amino acid, as these are having positively charged, because although it is having three charge, two are positive and one is negative, but one positive and one negative would cancel each other, and we will be having in the end net one positive charge. So due to this net one positive charge, this amino acid is positively charged amino acid. So now we would talk about the histidine. Histidine amino acid is having imidazole ring. This is the imidazole ring in its side chain. And one more important thing that I would say about this histidine, the histidine is one of the means only amino acid from the 20th amino acid of protein that can be found in protein as both acidic and basic form at physiological pH. So what is the physiological pH? pH 7 is the physiological pH. So in the body at 7 pH, histidine could be found either in acidic form or the means either it will be formed as neutral or as means charge form. Either it can be formed as a charge or uncharged at physiological pH. So why it happened we would be studying in next slide, but here you can have you just have to keep in your mind that histidine is one of the amino acid from all the 20th amino acid that could be found as neutral or in charge form at physiological pH. However, all the rest amino acid on the physiological pH, they can be means form uncharged, means only in one form. They may be either charged, either uncharged, either polar, either non-polar, like this. But histidine only one amino acid that can be formed in two form in a physiological pH. And due to this property of histidine, it involved in various enzyme catalyzed reactions. So we would be also studying about this enzyme or how this histidine take part in the various catalysis process in next, maybe in our next lecture, that would be based on the enzyme. But here you have to just keep in your mind that histidine is only one amino acid that could be found in two forms at physiological pH. Now we would be talk about the negatively charged amino acid. So those amino acids that are having negative charge in their side chain are called as negatively charged amino acid. So these are the two amino acid. One is the aspartate and one is the glutamate. So here we have to understand why these are the negatively charged amino acid. So why these are the negatively charged amino acid, although they are having two negative charge and one positive charge? Why? Because one positive charge and one negative charge would cancel each other, and in the end we will be having one net negative charge on overall on this amino acid. So due to this net one negative charge, this amino acid would be negatively charged. So these are two, one is aspartate, one is glutamate. So I said previously that asparagine and asparagine and glutamine, these are the amide of aspartate and glutamate. So this is the aspartate and this is the glutamate. And why we are saying this asparagine and this glutamine are the amide of this aspartate and glutamate. We can see here, if we focus on this structure, we can see here in the side chain, if we would talk about the aspartate, so we can see this, its structure, and then when we would see this, we can see there, although it is a structure of aspartate, but in the side chain it is having amide group. So due to this having amide group, the aspartate now it's called as asparagine because it is the amide of aspartate. So similarly glutamine is the amide of glutamate amino acid. So this was all about the all the types of amino acid. Now we would be studying about the physical and chemical properties of amino acid. And why we are studying about these physical and chemical properties of amino acid? Because to understand the physical and chemical properties of amino acid, we could be able to understand the function of protein. So because protein are made up of amino acid. So once we know about how amino acid can chemically and physically behave, so we could be easily able to analyze what type of a function a protein that is made up of these amino acid would be able. So one of the most important property of amino acid, we can say it is the aromaticity. What is this? Is the ability of absorption of light by aromatic amino acid. So the aromatic amino acid, one thing you have to clear in your mind here, that different amino acid having different chemical and physical properties. Okay, so one of the important property that is having that is exhibiting by aromatic amino acid is the absorption of ultraviolet light at 280 nanometer. So why this absorption? Why the absorption of ultraviolet light make unique this amino acid? Because first of all we would clear here that what amino acid, which amino acid absorb the light, ultraviolet light. So these are three amino acids that are aromatic amino acid. So we can see here, this is the wavelength and this is the molecular molar absorption, means absorption of the wavelength or absorption of the UV light. So magnitude of absorption you can say. So here we are seeing three amino acid, tryptophan, tyrosine and phenylalanine. So these all are the aromatic amino acid, and tryptophan and tyrosine are able to absorb UV light at 280 nanometer in more quantity or more extent as compared to this phenylalanine. So question is why? So answer is here, why it could be able for tyrosine and tryptophan to absorb light at UV in UV region or 280 nanometer. However, phenylalanine is not able to absorb such extent of light that tyrosine and tryptophan can absorb. So answer is here because tyrosine is having functional group, that is hydroxyl, that absorb light, and the tryptophan indole ring is having ability to absorb light at UV region. So due to the having this indole group and this hydroxyl group, these two functional group make them unique to absorb light at UV region. So due to this ability, tryptophan and tyrosine absorb light at 280 nanometer. So why this light absorption is important, and why this absorption of light make them unique? Because if these amino acids are present in the protein, so we can easily if these amino acids would be present in the protein and they are having the indole functional group and hydroxyl group that can be able to absorb light, so we can easily find out or identify a particular type of protein that is having these particular amino acid from an unknown mixture of protein. So what you understand, are you understanding what I wish to explain you, that why this absorption of light by amino acid are important and why this aromaticity or light absorption making them unique? Because just having an idea, we are having a solution of protein that are having multiple type of protein, and we have to find out one important one particular type of protein. For example, we are having a mixture of protein, and in this mixture of protein, someone give us a mixture of protein and he mix in this mixture one of the enzyme. For example, one of the protein from the you can say from one of the protein that is having one of the particular type of protein that is having a large number of aromatic amino acid, and this protein is named as protein A. So someone give us a mixture of protein that is having protein A to Z, and protein A is having large number of aromatic amino acid, and he told us that find out protein A from the sample, so and identify protein A from the sample. So how we could be able to identify protein A from the sample? When we will analyze it under spectroscopy, that is an instrument that identify all those protein that could be able to have these aromatic amino acid. So what will happen when we analyze this sample under spectroscopy? So the protein A that would be containing higher number of this amino acid, or high percentage of this aromatic amino acid, would be found to absorb UV light at 280 nanometer. So due to the absorption of light by protein A, we could be easily identify, yes, this protein A is having higher number of aromatic amino acid. So in short, you can just keep in your mind that due to this aromaticity or the ability of aromatic amino acid to absorb UV light enable us to functionally characterize protein or to identify a protein from unknown mixture of protein. So now another chemical property of amino acid is their chirality or chirality, you can say. So what is the chiral center? What is the chiral center, or what is the chirality? And this is carbon is having 4 valencies. Whenever in nature you will find carbon atom, you will find that carbon atom is having 4 valencies, means it will be having ability to bound with 4 different or 4 same other atoms. So in case of means amino acid, this carbon atom, that is the alpha carbon atom, in amino acid we call this carbon as alpha carbon atom. So in amino acid, this carbon atom will found to means bounded with 4 different other groups. So we can see here, one is the R group, one is the hydrogen atom, one is the NH3, and one is the amino group, and one is the carboxylic group. So when a carbon atom found to bind with 4 other atom, so this property is called as chirality, or the carbon atom or carbon center is a chiral. Or if a molecule is having carbon atom that is found to bound with 4 other atoms that are different from each other, so this molecule will be said as chiral molecule. Okay, but all the amino acid are chiral and having chiral center except the glycine. Why? Because glycine is not having 4 different atoms bounded with the alpha carbon, because glycine is having side chain that is having another hydrogen. So we can see here the glycine is having alpha carbon, and this alpha carbon bound with 2 same atom, that is hydrogen, and 2 different atom, one is amino group and one is carboxylic group. So except the glycine, from the 20 amino acid, all other amino acid is having chiral center, or all the amino acid are chiral. So due to this chirality, what happened? Amino acid can arrange itself into 2 stereoisomeric form. One is L form and one is D form. So what is this stereoisomer? I hope you all have been studying previously in the organic chemistry, I think, course, but I would explain here. The isomers are those molecule that are having same atom, but the atom are having different arrangement in space. So these are the isomer of each other. Or what are the stereoisomer? Stereoisomer are those molecule that are having all those same atom in its structure, but these are atom having different 3D structure or different arrangement of atom in this space. So due to this different arrangement of atom in the amino acid, why this arrangement is different? Because in amino acid, carbon atom is found to bound with different atoms, 4 different atoms. So these 4 different atoms can make bonding with two in 2 forms. So these we say as 2 stereoisomer of each amino acid, means each amino acid is having 2 stereoisomer. It can be formed in L form or it can be formed either D form, and all these forms are non-superimposable each other. So although those stereoisomer that cannot be superimposable or mirror images of superimposable each other, means not identical each other, are called as enantiomer. So the 2 stereoisomer of each amino acid are enantiomer. So and one thing more, that although amino acid can be exist in 2 stereoisomeric form, one is L form, one is D form, but in the protein only left-handed, that is L amino acid are present. Protein is not having D amino acid. So you have to keep in your mind that only the L form of a amino acid are present in the proteins. Now, and we would be studying another property of amino acid, that is optical activity. So the all those amino acid that can be exhibit to stereoisomeric form could be able to misrotate in plane polarized light. So the ability that enable amino acid to rotate in flow under plane polarized light is called as optical activity. So all those amino acid that can be rotate, that can be exhibit into two stereoisomeric form are optically active. So except glycine, because glycine cannot rotate in plane polarized light, and glycine is not optical active. So why glycine is not optical active and why glycine cannot means able to rotate in plane polarized light? Because glycine is not having L and D isomer. Why glycine is not having L and D isomer? Because glycine is not having 4 different atoms attached to the alpha carbon atom. So glycine is not having cannot be able to rotate in plane polarized light. So another property of amino acid, that amino acid can be exist at deuterine, and amino acid can be present as can be deuterine. So what are deuterines? Deuterines are those ions that are having both charge. So at physiological pH, so if we are having an amino acid, you can see here this is an amino acid, it is having a amino group, a carboxylic group, a hydrogen atom number 3rd valency, and an R group number 4 valency. So this amino group can be, amino acid can be having 2 ions or 2 charge at physiological pH, that is one is positive charge and one positive charge of amino group and the negative charge of carboxylic group. So due to this ability of having both charge at a time at physiological pH, so amino acid is called as deuterine or dipolar ion. How this different charge could be formed in amino acid we will be discussing in coming slide, but you can just keep in your mind that amino acid is having phenomena that is we call as dipolar ion or deuterine. And what are dipolar ion? A dipolar ion is a chemical species or ion that is having both charge as physiological pH. So this is one of the unique property of amino acid, that amino acid can be found to have both charge at physiological pH. And now here we would be studying that how this amino acid could be able to exist as dipolar ion or deuterine at physiological pH. So and this ability of having both charge at physiological pH enable amino acid to act as acid base or to exhibit acid base properties. So how amino acid could be able to act as acid or base means amino acid are having ability, they can be in a solution, they can be able to act as an acid one time, or another time they can be able to act as a base. So how it could be possible we will be seeing here. So here we can see that this is a this is a graph, and we are seeing here it is depicting the pH that is from lower pH to higher pH, 0 to 13. So pH from 0 to 7 it is the acidic pH, and from 7 is the neutral, and from 7 to 13 it is the basic pH. So what happened, at the acidic pH, that is the 0 pH, means extreme acidic pH, amino acid would be present in the solution as in the what form? In the so amino acid would be present as a protonated form. So what is this protonated form? Protonated form means amino acid would be having this proton in its structure at the acidic pH. So what are acid? Acid are the that chemical species that could be able to donate a proton. So at very acidic pH, amino acid would be having capability, would be having this proton and would be having capability to give this proton. So at the very low pH, amino acid would be present on the protonated form. And then what will happen when pH will increase? So what will happen? This proton this amino acid carboxylic group start losing its proton. So when the amino acid carboxylic group will lose its proton, so we would be having deuterine, that is amino acid that is having dipolar ion at physiological pH, means at pH 7 we would be find amino acid in this form having both ion. So it will be present, it will be found at neutral pH or pH near neutrality. So after that what happen? When we would be having, just you have to keep in mind, if we are having amino acid that is glycine and it is present in solution, so what will happen? This amino acid would start losing first its proton from carboxylic acid, and then what at extreme acidic pH, and what happened then? When the pH would be go towards neutral pH, so amino acid completely lost its proton, and we would be finding dipolar ion, that is deuterine, in solution at neutral pH. And then what happened then? When we will go towards the basic pH, then the proton from the amino group would start losing, and at extreme basic pH we would be having amino acid not having proton in the amino group. So here in this graph we also need to understand that this is the titration curve, and it is showing two phases of titration. Two means two we can say the two portion of titration. One is this portion from 0 to 7, and from 7 to 13 or 12 this is the other portion. So in the each portion at the midpoint of the titration curve, the pH would be equal to the pKa. So pH of the solution would be equal to the pKa. So what is the pKa? pKa is the ability of an acid to give up the proton. So pH at the midpoint of the titration, the pH would be equal to pKa. So and then the amino acid would be form an equimolar concentration of two species. So one the starting when we start the titration, so the starting species was this form, and then at this point the final species at pH 7, the final species was this form, but this pKa1 is the middle form, and this middle is the middle point, and is this middle point at pKa 2.34, that would be the equal to the pH, that is 2.34. We would be find an equimolar concentration of this form and this form, and it will means proceed similarly at this stage, that it's at the middle of the titration curve of the second stage. The pKa would be equal to the pH, and we would be finding the equimolar concentration of these two species at this midpoint of the titration curve. So what is this pi? Pi is the point, or pi is the pH where the amino acid is having no net charge. So what would be the species that would be present at pi? This would be, because I explained you previously that although when an amino acid is having two charge, one is positive and one is negative, so these two charges cancel with each other, and amino acid would not be having any net charge. So at pi is the point where amino acid, pi is the point, or pi is the pH where amino acid would not be having no net charge. Amino acid would be found as a neutral. So pi is the pH where amino acid form in the neutral form. Now why we have to understand that why and pi is also called as isosteric point. Pi is called as isosteric point. Isosteric point is a point or is a pH at that the amino acid would not be having any net charge. So why this isosteric point is important? This isosteric point is important for protein to isolate. So just assume that we are having a protein, and this protein is having amino acid, and these amino acid and when we put this protein that is having amino acid in the solution, and then amino acid is not having any net charge, so this would help us to isolate protein. Because how isolate protein, how we can isolate protein on the basis of this charge? So you have to, or by understanding this titration curve, so you understand just this, that that we have to isolate. We are provided with a solution of protein, and protein is having different amino acid in that. So we wish to isolate the protein. So how we would isolate? What we do, we put this protein in a solution, and when the pH of the solution would be higher, so what charge pH of the solution would be acidic. So at that time protein would be having positive charge. Means why positive charge? Means pH, protein would be having positive charge when protein would be having the positive charge at the pH that would be higher than pi. So what would be happen then? If protein would be having the positive charge, or amino acid would be having positive charge, so the positive charge protein would be moved towards the negative charge electrode, that is cathode, and the negative charge protein would be moved towards positive charge electrode, that is anode. So actually this protein would be isolated via process of electrophoresis. I think this is I don't wish to explain you lot more about the electrophoresis, but you have to just keep it in your mind. The protein can be isolated via the process of electrophoresis, gel electrophoresis, on the basis of the charge that the amino acid of protein are having. So how we do this process? We just put the protein that is having amino acid in the solution, and we just change the pH of the amino acid. So by changing the pH of amino acid, by changing the pH of the solution, amino acid will be exhibiting different charge. So when that amino acid have exhibiting positive charge, so the protein would be moved towards the cathode, negative charge electrode, and when the amino acid would be having negative charge, the amino acid would be moving towards anode, that is positive charge. So you just have to keep in mind that the charges on amino acid would enable protein to isolate from a mixture of multiple protein. Via process, we term this process as electrophoresis. So now we can here we need to understand that how we can calculate isoelectric point of amino acid. So isoelectric point of the amino acid can be calculated by combining its pKa. So here we can see that we are having titration curve of glycine, and glycine is having 2 pKa, that is pKa1, one is pKa1 and one is pKa2. So by combining these 2 pKa and dividing with 2, we can find out the pi of amino acid. So you can maybe have this question in your exam, that how if you would be provided pKa of 2 pKa of an amino acid, and you would be asked to calculate the pi. So what you have to do in this case, you have to just combine the pKa of 2 amino acid and then that are having non-ionizable R group, and then have to divide with 2. So these was about the chemical and physical properties of amino acid. Now we will be studying a little bit about how amino acid can be metabolized. So actually amino acid metabolize via in your liver, and via cycle that we say as a urea cycle. So what happened, just keep it in your mind that if you are ingesting protein from your food, so then first protein would be digested in your mouth, then it will be absorbed in your intestine, and from intestine it will first protein would be digested into smaller peptide in your stomach, in your mouth and stomach, and from smaller peptide the amino acid would be released in the intestine and then absorbed by intestinal cell and then transported into the liver, and in liver mitochondria the amino acid metabolism would take place, and amino acid in liver this liver mitochondria would be metabolized and convert into urea via cycle that we call as ornithine cycle or urea cycle. So you for understanding more about the urea cycle, you can also watch my another lecture that is whole basing on the urea cycle. So here we see that how these amino acids join together and form the protein or polypeptide chain. So up till now we studied that what are amino acid, what are their physical and chemical properties, and now we are seeing that how protein form from these monomeric unit that are amino acid. So actually what is this protein? We are having a picture of protein here, a diagram of protein. So what is protein? As we can see here from this diagram, actually protein are the polymer of these amino acid. So you can see here this little monomeric amino acid join with each other and make a long chain, and we say this long chain as a polypeptide chain. So how this amino acid and this many polypeptide chain will join with each other and form a protein. So how this amino acid would join with each other, and how this polypeptide chain form, now we would be studying. So we are seeing here a simple diagram, that is the diagram of the amino acid. So we can see here, this is the alpha carbon that is attached with a hydrogen, and that is attached with a side chain, and one amino group and one carboxylic group. So what happened, this many different amino acid join with each other via peptide bond, that we call as peptide bond, and make a long polypeptide chain, and many different polypeptide chain join with each other and form a protein. So why we say this as a polypeptide chain? Because this is having multiple peptide bond. So due to having multiple peptide bond between the amino acid, we say this chain as a polypeptide chain. So how this polypeptide chain form, and how protein form? So this is the this is the first step of protein formation. How protein form from amino acid? Amino acid are simple monomer, and when this simple monomer join with each other via peptide bond, form polymer that we say as a protein. Actually protein are the polymer of simple monomeric amino acid. So all these simple monomeric amino acid join with each other and form a polymer of amino acid that we say as protein. So actually what happened, we are having here two amino acid. So we can see here, just take a glimpse, that you are having carboxylic group. Here you are having alpha carbon atom that is attached with hydrogen, that is attached with another R group, and that is attached with amino group. So this is one amino acid, and this is the second amino acid. So what happened, how amino acid join together and form polypeptide chain and then protein. So what happened, hydrogen atom of amino group of one amino acid and the hydroxyl group of carboxylic group of another amino acid will remove from these amino acid, and this hydrogen will combine with hydroxyl and form a water. And what happened then, this bond will make joining with this bond. So what we will be having at the end, we will be having a peptide bond. You can see here that C double bond O bond N and H. This is the peptide bond. So what happened, these two amino acid join with each other via single peptide bond. So this happened with again and again with many amino acid, and then many amino acid join with each other via multiple peptide bond and form a polypeptide chain. So here, and a polypeptide chain is having multiple peptide bond. So here we can see that what is the amino acid residue, what is N terminus and C terminus of polypeptide chain. So you have clear idea that what is the polypeptide chain. Actually the polypeptide chain is this one chain. This is having multiple amino acid that are joined with each other with multiple peptide bond. And how this peptide bond form? This peptide bond are formed via the removal of one hydrogen from amino group and hydroxyl from carboxyl group of other amino acid. And this reaction is called as condensation reaction, and this is the dehydration reaction, because when a water molecule removed from a reaction, so this reaction is called as dehydration reaction. So now one more thing, that when what is the amino acid residues? Amino acid residue actually is the leftover of amino acid after the removal of hydroxyl from carboxylic group and ammonia and hydrogen from amino group. So if you are having an amino acid that is not having hydroxyl group in carboxylic group and not having means hydrogen atom in amino group, so these are called as residues. So these amino acid residues, after the removal of hydroxyl and after the removal of hydrogen via the condensation reaction or the dehydration, would join with each other via peptide bond and will make polypeptide chain. So the group that is having amino group, so the amino acid or the polypeptide chain, that side of the polypeptide chain that is having free amino group at the end will call as N terminus, and the polypeptide chain that is having free carboxylic group at the end would be say as C terminus. So this N terminus and C terminus are very important terminus because these are important for the biological activity of protein, and we would discuss about these terminus in our further lecture. But here you have to just keep in your mind that each polypeptide chain made up of amino acids after the joining of amino acid via the peptide bond, and a polypeptide chain is having two terminus, one is C terminus and one is N terminus. So we always read this any polypeptide chain from its N terminus to C terminus. Okay, so what does it mean by reading? If I will say that I am having a polypeptide chain and this polypeptide chain is having multiple amino acid, so kindly write down the name of this polypeptide chain. So from where you have to start? You have to start from N terminus. You will write first the name of that amino acid that is present at the N terminus, and then you will write the second name of second amino acid, and then so on till C terminus. So one more thing that I would wish to clear you here, that when two amino acid, here we can see that when two amino acid join with each other, so they form one peptide bond. So they form a one peptide bond, but we say this peptide as a diapeptide. Why? Because it is having two amino acid. Okay, so if 5 amino acid will join with each other, so it will be having 4 peptide bond, and it will called as pentapeptide because it is having 5 amino acid. So peptide bond number is given by the number of amino acid. Peptide bond number means pentapeptide, tetrapeptide. Tetrapeptide means 4 amino acid that join with each other via 3 peptide bond. Okay, so peptide bond always number with the number of amino acid. So hexapeptide means there are hexapeptide. What does mean by hexapeptide? Hexapeptide means there are 5 peptide bond that join 6 amino acid. So this is so on. We will say like this. It could be come in your exam that write down the someone can give you the means polypeptide chain and would say that write down how many peptide bond are present in the polypeptide chain. So you have to understand that if there are the 4 polypeptide peptide bonds, you can say 4 peptide bond. So it means that it is pentapeptide. Okay, 5 amino acid join with each other with 4 peptide bond. So this was all about our today's lecture. I hope you enjoy this lecture, and now for further reading you can consult with this book. One is Steyer and one is Larringer, and I would specifically refer you the Larringer because I also in love with Larringer because all these topics are very much elaborated and easily present in the laryngeal. And in our coming lecture we would be studying that what is the structure of protein. So here till now we understand that what are the amino acid and how amino acid assemble together and form the protein. And now in the next lecture we would be studying about the structure of protein. So thank you very much for your understanding. Allah Hafiz.